The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, this is episode 98 of eLife's podcast, and I'm Chris Smith from The Naked Scientists. In this episode, why it's a myth that only the colourful frogs are the poisonous ones, the researcher using movies to probe baby brain development, and a new technique to match the cancer drugs with the best prospects of working to a patient's disease. But first, does an extra year in school change your brain? In 1972, a pint of milk cost 6p and a litre of petrol, leaded of course, was only 8p. Weren't those the days? But in that same year, when Alice Cooper appropriately released the hit song School's Out, an important change happened in the UK education system. The school leaving age was upped by a year, from 15 to 16, and this presents scientists with an opportunity. If education boosts brain power, which it arguably must, then what would we see if we compared the brains of broadly similar people who either did or didn't benefit from that extra year in school? That was what Nick Judd and Roger Kivitt at Radboud University wanted to know. Nick first.
We know that education is one of the largest positive impacts out there. It's a major contributor to socioeconomic disparity, and it's correlated to almost every positive life outcome. So we were really interested in if it had effects on long-term structural changes in the brain. So we looked at around 6 different structural brain metrics. So how big your brain is, how thick your cortex is, the surface area of your cortex, also the volume, fractional anisotropy, which is pretty much just how white matter diffuses in the brain. We pretty much took an approach to try to look at almost every structural property.
And Roger, what was your approach here? Where did you get the data from?
Yes, so we used a very large project from the UK called Biobank, which has hundreds of thousands of people in it. And we used the fact that in the UK there was a law change in the 70s where people were forced to go to school for an additional year. So before, every child had to go to school for at least 15 years, and after that for 16 years. So we could use that change in the law to compare children who were born just to one side of that cutoff, so they went to school at least 15 years, to children who were born just after that cutoff, which meant they had to stay in school for an additional year. So we could use that data to look at their brains years later and compare the people who only differed in how long they had to go to school.
That's quite a considerable intervention, isn't it? An extra year of education. So if something, I suppose your rationale is if something's going to make a difference, this will be it.
Yes, so for years, people have used brain training games for sometimes minutes or hours in the hope that that would yield long-lasting consequences. So we thought if you go to school for a whole additional year, we might stand a chance of seeing something in the brain, even a few decades later.
And what do you find, Nick? Is there an impact of doing an extra year of school?
Yeah, so we didn't find an effect on any of our measures. So we also looked at different regions of the brain, and we didn't find any effect on any structural brain measures. A key distinguishing part of our approach to prior research is this natural experimental approach, which allowed us to really isolate and tease apart education in itself from all of the societal factors that might lead some people to receive more education than others.
You must have been a bit disappointed, Roger, when you got that result. You must think, well, actually, we were hoping we were going to show this dramatic difference. You saw nothing.
Well, we tried to be dispassionate and not be too disappointed, but we also thought it's interesting precisely because we do know that education does have long-term benefits on your cognitive ability, so how well you can remember things or think about problems, and on health. So we know that this educational intervention did have an impact in these same people we looked at, but we just weren't sure whether the impacts would be in the brain in the long run. And the way we thought of it is maybe it might be like going to the gym. That might bulk up your muscles for a few weeks or days or months, but maybe if you don't use those same muscles for a long time, then maybe 20, 30 years later, you don't see the same impact.
We've just seen a paper in Science Advances where a German study has looked longitudinally at, albeit a relatively small group of people, but they find people doing cognitively demanding jobs, which appear to stretch the participant, do appear to maintain their cognition for longer than people doing less demanding jobs. So do you think that the education just gives you access to the lifestyle that gives you the sorts of benefits, Nick, that you're saying accrues through education? Or is it something else that's going on here?
Just to clarify, the effect of education on IQ and other cognitive abilities is pretty well established with natural experimental designs. So it's really, we don't pick it up in the brain, but the beneficial effect of education on cognition is there.
Do you have a reason for why that might be, Roger? Can you speculate as to why it can have these profound effects, but it's not reflected in the brain? Are we just not looking hard enough, or is the resolution of our imaging not good enough to pick up something that's probably a bit too subtle?
Yeah, so those are a few of the things we've been discussing. One thing we would have liked to go back in time and study people's brains right after they had the additional year of education, because that would be the best comparison to see right when the children spent an additional year in the schools, do we see effects on their brains at that moment? Because maybe there was an effect initially and then it essentially faded out. And there's some ideas that your brain might have this short-term reorganization that is visible. And the way we think of it is like roadworks. So there's a lot of noise and mess. But after a while, it reconsolidates. So we call that renormalization. So your brain goes back to normal and it's almost as if nothing ever happened. But those benefits for your brain are still there. They're just not there at the level we can pick up with an MRI. So the other possibility is that using a different scan or looking at functional activation, that we might find these traces of the effect there. But given how much people use this exact MRI technique, we thought it was valuable to see, can we find these effects using this type of scanner?
The take-home messages, what do you think is really the bottom line here then?
Well, the bottom line, I think, is that to understand how the brain develops, we have to study it developing. And that sounds a bit obvious, but for a long time, we only had the techniques and the money and the resources to study one brain once. So you come in, you get scanned. But increasingly, we're realizing to really tease apart this puzzle of how our brains develop and how they allow us to do all these wonderful things, we need to see how your brain changes over time. So more and more neuroscientists are scanning people, say, every year while their life unfolds. And I think that's going to give us a lot more new insights over the next years and decades.
Indeed it will. Roger Kivett and Nick Judd there. Next, how did frogs become toxic? Although they were first documented scientifically in the late 1700s by the French biologist Georges Cuvier, brightly coloured poison dart frogs were known for centuries before this by native South Americans, who laced the tips of their blow darts with the animal's toxic skin secretions, hence the name. But are the colourful frogs the only toxic ones in the family, as is widely believed, or are their duller coloured brown cousins also capable of packing a chemical punch? That is what Rebecca Tarvin from the University of California at Berkeley has been looking into.
We are very interested in understanding how frogs become toxic. Most people have focused on brightly colored frogs and finding out how toxic they are, but very little focus had been put on the brown frogs. So we actually didn't know how frogs transition from brown to brightly colored and from non-toxic to toxic.
Where do the frogs come by their toxins?
Poison frogs and other animals that have this trait, they get their toxins from what they eat. Things like mites and ants and occasional beetles or fly larvae, things like that. And those arthropods have lots of different chemicals. Some of them you might call like a toxin bomb. Some of these mites are referred to that way. Ants also have a really broad diversity of compounds. Most of these toxins are not even studied. We don't actually know what the effect is on an animal and how toxic they are. They could just be distasteful, which is also useful, to deter predators. You don't have to kill a predator to keep it from eating you.
I suppose there's a number of different problems to surmount then if the frogs want, in inverted commas, to do this, because not only have they got to not die from the toxin themselves, they've got to be able to separate it from the useful things they do want to digest and then put it somewhere in their body and then keep it there so that it's useful. That's a lot of evolutionary steps.
That's right. It's quite complicated, and actually we have very little understanding of how it happens aside from it does happen. So there's a couple research groups, including my own, where we're really interested in trying to understand how exactly the toxin goes from even starting at the mouth or the stomach, how exactly it gets into the skin, because that's where it's present in really high concentrations.
And what, by studying the darker, uncolourful brown frogs and asking how are they different from the bright colourful ones, despite being very closely related, you're hoping that there'll be some of those mechanisms disclosed.
Yes, that's right. So if you want to understand the evolution of any sort of trait like this, you need to kind of be able to compare animals that have that trait with animals that don't. And so that's where these brown frogs come in. We didn't previously know if they actually lacked this trait because there's a sort of bias. People look in the wild and they see these brightly colored animals and they think, I wonder if that species is toxic. So they go and they collect a sample and show whether or not it has toxins. But people, not surprisingly, aren't as curious about the brown ones. So there was a huge oversight in the literature about what these animals were doing.
And are the brown ones toxic?
They do seem to have alkaloids, which are the compounds we find in the colorful frogs, at levels that are clearly measurable. In some cases, I would think it has an effect on their predators, but in a lot of cases, they have quite low levels. So it's something that's detectable, but not necessarily toxic towards predators, although we actually don't know that.
These brown frogs, then, they are a step towards becoming brightly coloured, highly poison, colourful frogs, but not the whole hog. So have they got some of the right apparatus, but not all of it then, to move down that evolutionary path?
That's what we think. So before our study, there was an assumption that the main difference driving whether or not a frog was toxic was what they were eating. And that's because we know that they get their toxins from their food. They actually can't synthesize any of these toxins. And so people assume that, well, these toxic frogs, they must be eating a different diet. And in our study, we show that actually they eat a lot of the same arthropods, same mites and ants. And so it's not just diet that's driving this. There's something else different about the physiology of those accumulators, the sort of colorful species.
Apart from busting a myth that brown frogs are not taking on board some of these toxins and maintaining them, at least at a low level, what does this study add then? Where next with this?
The question we set out to answer was, how do frogs accumulate toxins? And in order to do this, we have to understand not just the brightly colored frogs that do accumulate toxins, but their close relatives that aren't as toxic or as colorful. And so what we're working on now is more direct tests of this, where we take a known quantity of an alkaloid and feed it to a colorful frog that we know sequesters it very well, and also feed it to a relative, a dull-colored frog that in the wild doesn't show as high of an ability to sequester the toxin. And then we can compare, what happens to that toxin? Does it reach the skin? What quantity of the amount that we give them reaches the skin, and how does that differ? So we're starting to dig deeper into these mechanisms.
Rebecca Tarvin, who's studying the evolution of the bright colours and toxic disposition of poison dart frogs. This is the eLife podcast from The Naked Scientists, and I'm Chris Smith. In a minute, the parasite that manipulates a plant and an insect, acting like a dating agency in the process to facilitate its spread. Intrigued? Stay tuned. Before that, though, there's an old adage, never work with children or animals, because it's known to be notoriously difficult, especially when it comes to certain aspects of science and wanting to generate high-quality data from repeated measurements. And that's where the kids can be particularly tricky. But Cameron Ellis at Stanford University has partly solved the problem with the help of movies. He's interested in how the developing human brain wires itself up and responds to different experiences and stimuli over the initial months of life. When it comes to the visual system, the current gold standard for doing this is to present various defined shapes and colours and monitor the response. But it can be very hard to keep subjects still and motivated to remain engaged, and this can mar the quality of the data. So can movies, designed as they are to be compelling and immersive, be used instead, he wondered.
It's a real curiosity as to why it is that we are born immature. If you look at horses, they can walk a few minutes, maybe an hour after birth, whereas we take 9 to 12 months to learn how to walk. The same is true for our visual system as well. Although other animals might have a relatively mature visual system, meaning that the way in which they process the visual input that they receive is similar in infancy as it is to when they are an adult. That's not the case in humans. Our visual system is really different. Why it's different is a curious question that we don't fully know the answer to. But one explanation is that in order to acquire the kind of expertise that we use for processing the visual world in our adulthood, that can't just all be baked in by our genetics. Instead, some of that has to happen as learning during our lifetime. And so we need to learn how to see the world, and that's part of what's happening in that first year of life.
Are
Yeah.
you getting at that there's a rough map there, that things seem to be wired up roughly right, but what we need is the icing on the cake, the finessing of that system in order to get the amazing visual system that humans have when they're older?
Yeah, so when we started this work about 5 years ago, we didn't know whether the macro structure or the general organization of the visual system in infants in place. And when we found that that was very surprising, that in general, the big organization that is present, at least in parts of the way that we process vision, is very similar between infants and adults. But despite that similarity in the big stuff, we still think that over the course of development, there's lots of changes in the little stuff, the perfecting of the fine-grained details.
So what does the bringing movies to bear on the problem buy you?
Movies are a really amazing stimulus for showing infants as well as other populations like patients who otherwise might be uncomfortable in the kind of neuroimaging environment that we use to measure brain activity. The reason is that movies are naturally engaging. And so when you use a very engaging stimulus, you're likely to get more data that is low emotion. That is something that we have seen in our data, that infants have more attention and they will move less when watching movies. And so it really comes down to a data quality issue in one sense, that we want to see kind of how good the data can be when using movies. But there's another reason for using movies as well, is that it's really nice to see that the brain's response to somewhat naturalistic stimuli, I don't mean that they're fully naturalistic because it is still watching a movie that was designed by a director and what have you, but somewhat more natural, kind of everyday type experiences, evoking brain activity looks something like the kind of brain activity that we identify when we're using kind of a more of a test tube and beaker approach, where we are showing specific shapes with specific timing that are well tested on adults and other infants to validate that they reveal certain aspects of the brain.
And do your results align when you show movies with when you've done other rougher probes using artificial stimuli like shapes and so on? Do you get broadly the same results?
When we use these really well-controlled experimental stimuli like colorful shapes, those methods are considered the gold standard of identifying the visual organization in the adult brain as well as we think in the infant brain. But when we use movies, we can get rough approximations of those same maps from measuring the brain's response to the movie data. So this tells us that the kind of way in which the infant brain is processing movies recapitulates or recreates the structure that we could have otherwise seen by using these precisely controlled experimental stimuli.
Which must give you confidence for a start, but also mean that because you can vary what's in the movie, you can actually quite easily adapt this and probe things slightly differently, but also do it repeatedly because you've got really good engagement from the kids in the scanner. They want to watch the movie.
That's exactly right. Beyond it being a proof that movies are a great way of evoking brain responses in these kind of messy situations, this is also a way of showing that movies are a really powerful tool that ought to be deployed and used in many different cases for studying the infant brain. For instance, you can use movies to naturally compare how how infants' brains respond to different languages, or you can use movies to test how they understand social events and social interactions. You can use movies to study how infants are reasoning about others' minds. And we think that this is just generally a really good demonstration that movies ought to be a big part of experimentalist toolkit when studying the infant mind.
I was just going to say, what will you go on now? Now you've given yourself the confidence to pursue this, what will you go on to test or explore with movies, because it will be easier to do that?
One of the exciting ways in which we're using movies right now is that we're using movies as a way to study the infant's brain response to language. doing an experiment where we're studying how the infant brain responds to familiar languages like English for the children that speak English versus unfamiliar languages like Japanese for the children that don't speak Japanese. And we want to see to what extent the infant brains differ when they're hearing English versus Japanese. Another really exciting method that this paper gave us confidence we could pursue is that we can use movies as a way to find out directly what is similar between infants and adult brains. We can ask, how does the infant brain process this movie, and how is it similar or different to the way that an adult brain processes this movie? And that could be a really effective and unique way of asking this question: what makes infants unique? How do their experiences differ from adults' experiences?
Cameron Ellis from Stanford University. Next comes a tale of deception, seduction, and sexual exploitation, and it's all being done by a parasitic bacterium called phytoplasma. This infects plants, into which it secretes a protein called sap54. This turns flowers into leaves, making the plant a superior food source for leafhopper insects, which spread the bacteria when they come to feed. But even more incredibly, that protein also makes the plant even more attractive to females if there are males of that insect species present, further reinforcing the population of vectors who can spread the infection. As Zygmunt Orlovskis, who's from the Latvian Biomedical Research and Study Centre and unpicked all of this, puts it, it's effectively operating like a molecular matchmaker.
Phytoplasmas are parasitic bacteria that infect plants. They are transmitted by insects. So when the insect feeds in a plant, it picks up this bacterium and transmits to another plant, very much like malaria parasites, which are transmitted by mosquitoes. One of the features that this bacterium does to plants is it changes plant development, particularly turns flowers into leaf-like structures. You might be wondering, why does it do that? Well, one hypothesis that we had was that these manipulated leaf-like structures could be more attractive to the insect vectors. So we went on to test that first. And it turned out that actually these leaf-like structures are not required for increased insect attraction to these plants.
If the plant doesn't make flowers anymore and it makes more leaves, does that render it sterile? And is the possible reason it's doing this just to make the plant grow itself to death, so there's loads and loads of plant tissue to feed loads and loads of parasites?
Yes, that's an obvious question, and that also puzzled us. So the plant indeed is turned sterile and cannot reproduce anymore. So the question is, why would the parasite want to do that, to kill its own host, unless it can facilitate its own spread before the plant actually comes to senescence and death?
Do you think that's what's happening then? Is the bacterium making the plant sterile so that it just becomes basically a bacterial factory feeding lots of vectors, which will make sure that that parasite spreads?
Yes, indeed. That's one potential explanation what happens in this system.
And have you got to the bottom of how it's doing that? Do you know what the signal is that the bacteria release that make the plant respond in this way?
So the bacterium actually secretes a particular protein into the plant. And this protein is changing not only plant floral biology, but it's also changing processes in the leaves, rendering the plant more attractive to the insect vectors. But it does in a very peculiar way. It primarily biases female insects towards plants that are producing this bacterial protein compared to plants that lack this protein.
The protein itself, perhaps you could tell us what that is in a second, but the bacteria make it. It has this effect on the plant to convert flowers into leaves to make basically more food for parasites, we hypothesize, but it also in the process makes the plant more attractive to female vectors, the insects that are going to spread the parasite.
Yes, and curiously, bacterium needs also the plant exposure to male insects and production of this protein inside the plants to make the host more attractive to females for reproduction.
It's incredible. It's so intertwined, isn't it? And so that basically it's integrating all these different signals. How does it know the males are there? How does the parasite know that the male insects are there to start with, to tell the plant to lure in more females?
Male and female insects produce different signals or elicitors, and plant mounts different responses to both of them. While males and females both feed on the plant, only females lay eggs, and potentially the egg-laying stimulus creates some additional responses that the males do not have. So when we compare male and female exposed plant, which is producing this protein SAP54, and compare how other females choose between these plants, we see that you require male exposure of leaves that produce SAP54 for the females to come and lay eggs.
How on earth did this evolve in the first place? It's so complicated. You've got a bacterial parasite secreting an effector that does things to the plant to change its behaviour to different behaviours in response to males and females of an insect species. And it seems almost impossible to believe it evolved.
Yes, it is a very remarkable adaptation, and the work in many other labs, including Philippe Raymond's group in Switzerland, has demonstrated that insect eggs themselves contain elicitors in the eggshells that can trigger responses in plants that are sometimes very similar to bacterial infections as well. So the plant has evolved to recognize the different stimuli and elicitors that it can find and confront in nature. And it seems that this bacterium is somehow hijacking the insect sex-specific responses to achieve its manipulation of plant leaves and make them more attractive to female insect vectors.
But it's also manipulating the insects because it lures in more of the opposite sex. It's like kind of a dating app for vectors, isn't it? And it's drawing them in when there's the most to be gained through having them there mating.
Yes. Hence, we also are thinking of the system, it's kind of analogous of a molecular matchmaker to make male exposed plants more attractive to females. So it's a very interesting phenomenon.
An incredible and intricate story, and all from one bacterium too. Zygmunt Orlovskis there. Cancer is a genetic disease, and different cancers are driven by different constellations of genetic changes. And if we read those changes, in some cases we can make predictions about the likely aggressiveness of a tumour, and also, critically, which treatments might produce the best responses. A good example is breast cancer, where some tumours express estrogen receptors that promote growth, but are also an opportunity for intervention. Now, with her system called Retriever, the University of Oslo's Marika Kuja is trying to go a step further. By picking apart a patient's cancer, cell by cell, and reading the patterns of gene expression being displayed, and then comparing those patterns to similar profiles obtained from lab-grown cancer cell lines with known responses to drugs, it's possible to make highly accurate predictions about what treatments are the best fit for the individual's disease. It's a further step towards true personalised medicine.
So we basically developed a computational tool that we call Retriever, and it basically works on starting with this tumor sample, where we specifically look at what is happening in each individual cancer cell. This makes it possible for us to look at, for example, which genes are active or inactive. Now, we compared these profiles to similar profiles available from a very large database where cancer cell lines had already been tested with different drugs. Now, the idea of our method Retriever is that we can actually match these tumor profiles that tell us something about whether the tumor relies on specific pathways to survive and grow, and then look for specific drugs that disrupt those pathways in cancer cells from the same tumor type. So essentially what we're trying to do is attack what makes the cancer aggressive.
So in other words, because someone has already, in a dish effectively, interrogated cell types of the right sort and said, well, this gene is on, this gene is off, and this drug might or might not work, you've got a way of using that as a source of reference data and then applying the patient's individual case to that to extract what probably looks like the right combination of drugs.
yeah, that's exactly how it works. Yeah.
So how do you know then that when you put those drugs together, they're going to have the desired effect? Because obviously if they've only tested in the reference samples the drugs and the genes in isolation, how do we know that when we put them in combination, we get the outcome we want?
Yes, we did this through computational inference. So with computer models, we tried to test whether two drugs together might work in attacking these pathways. But what we can then also do, and we also did this in our study, is actually test them in the lab. So we basically have some cell lines and we treat them with drug combinations to see if they actually kill the cancer cells. And we saw that for 2 out of 3 cell lines that we actually tested, this was the case.
So it does appear to work when you try this. If you do this against real world samples, you do appear to come up with good combinations that are the best fit for that particular disease type.
Correct, yeah.
Have you actually tried this in real patients yet?
We have not, yeah. And this is really, really the next step.
And of course, the difference between things we grow in the lab, which are subject to the selection pressure of a dish, and a person's real disease, is that cancer is a really heterogeneous condition. A person's tumour has got a whole mixed bag of different cells and mutations in it, and an evolution built into the cancer. So do you think you're capturing that cross-section with this, or is that one possible shortcoming? You might be going for one bit of the tumour, but there are bits of it that you might be missing.
Partly, yes. So the thing that we can do something with is the actual tumor sample. So when we have the actual tumor samples, the type of data that we actually analyze is called single cell data. So we actually are able to look what happens in each individual cell. And what we do with this retriever method is we try to extract what is happening in the cancer cells. And we can even zoom into specific cancer cells that are very, very aggressive. For example, in our case, the cancer cells that do not express any receptors that can be treated with known drugs. And then we can use that information on those specific cancer cells, because that's actually what we're trying to kill, right? We're trying to kill the cancer cells. But the limitation of this is, of course, that we develop our computational tool based on experiments that have been done on these cell lines. So these have been grown in the lab. And there's definitely a big gap of then understanding whether the drugs that we predict to be working actually might work on the tumor itself, as the tumor is such a more complex and dynamic environment. It might be, for example, that other cells from the immune system or cells in the tumor microenvironment might play a very big role in, for example, not making these drugs work.
And what about other tumours? Presumably this could be extrapolated, this approach, to other tumour types.
Absolutely, yeah. So the database that we use for making these predictions actually has enough information available for, I believe it's around 13 different tumor types. So in principle, we could directly apply this method to other tumor types as well. But of course, the data that have been generated are usually based on cells that are very well known and very well studied, and those often come from cancer types that are more prevalent in the population, such as breast cancer or prostate cancer, or, for example, skin cancer. But there are also other more rare cancer types where we do not yet have this data available.
What you're describing here is really the dream of personalised medicine, isn't it? Something that people have always said, we want to tailor the treatment to a patient's individual disease. And the critics of that have always said, this is incredibly expensive and we just don't have the resources to do this. Were you to scale this to population level use, this is the kind of thing that Britain's National Health Service would start to offer as part of our breast intervention programme, for example. Is it realisable and realistic to try and do this, or is it so expensive and so labour intensive it will still be beyond the reach of healthcare providers?
I think it is possible with a tweak. So what we've done in this specific study is we specifically looked at individual cells, and we specifically studied the profiles that are aggressive in individual cells. But there's a method that's called bulk sequencing that can also be applied to our tool. It's not going to be as specific, so then we will have a mixture of cancer cells and immune cells, and we cannot really specifically zoom into the cancer cells that are most aggressive, but we still have a rough estimate of the profiles that are active and inactive and in cancer in general. So that is something I think that within the next 5 years, potentially use in healthcare.
That's an exciting prospect, isn't it? Marika Kucher there. That's where we have to leave it for this episode. You've been listening to the eLife podcast that's produced by The Naked Scientists. There are previous editions, the references, and also the full text transcripts for these programmes, as well as details on how to subscribe to this podcast in the unlikely event you don't already at nakedscientist.com/eLife. Incidentally, The Naked Scientists also publish a weekly science programme that covers the latest breaking science news stories, and you can find that at nakedscientist.com/podcast. From me, Chris Smith, thank you for listening. See you next time for episode 99. Until then, goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.